Oh, hi. So are the what? Oh, the alpha helix? Well, I don't know a lot about the alpha helix, but I know someone who does. So let me get him for you, okay?
Hi, I'm Dr. Jurgenson. I'm a biochemistry professor at Delta State, and I'm going to talk to you today about the alpha helix. So this is a model. It's a ball and stick model of the alpha helix, and I'm going to describe some of its structural features. So the way we describe proteins is they have a start point and an end point. Call it the N terminus and the C terminus, and that comes from the fact that the first amino acid residue that's made has a nitrogen atom that starts off the structure of the protein polymer, and it ends with a carboxylate group. So this amino acid chain of alpha helical amino acid residue chain has a specific orientation. So if I hold it this way or this way, it's actually different. And so the way we can tell the difference is by looking at the sp3 hybridized carbon that has the R group attached to it. So the way we start with this is by recognizing where the beginning is, so just start at the bottom. And then the alpha helix has this first residue. This is an sp3 hybridized carbon, and the side chain that's on it is a methyl group. And so the methyl group is a carbon, three hydrogen atoms, and it's pointing down. So as we go through the alpha helix from the N to C terminus, the side chains are pointing in the downward direction. So that's how I know we're going from the beginning, the N terminal amino acid residue, to the C terminal one on top. The model of this alpha helix also has different colored atoms. So black atoms represent carbon. The white balls, or the black balls, represent carbon. The white balls represent hydrogen. The blue balls represent nitrogen, and the red balls represent oxygen. So every single amino acid residue has at least some of those atoms in them. Some are larger than others. What we also see for every single amino acid residue is there's an amide bond, and the amide bond is shown here with the oxygen, carbon, nitrogen, and hydrogen. The bond between carbon and nitrogen is sp2 hybridized, and that allows for, it has sp2-like hybridization features, which allow for this to be a rigid structure. And so whenever we see the oxygen, carbon, nitrogen, and hydrogen together, we consider this to be a plane. It's flat. There isn't free rotation about the carbon and nitrogen. And that matters because if there was, this overall structure would be a lot more floppy. So we see oxygen, carbon, nitrogen, and hydrogen here. And as we move up the alpha helix, we see it again here, and we see it again here. And it's present in every single one of the amino acid residues that we see here because that amide bond is a peptide bond, is what we call it in protein structure. And it's one of the key things that holds this structure together and makes sure that it's rigid and that it's stable enough to maintain its structure so the protein can maintain its function. Something else to point out is this weird looking series of balls that are connected here. What this is actually the hydrogen bonding interactions that occur between amino acid residues. There's a hydrogen bond between this oxygen atom that's attached to carbon atom and then a hydrogen atom that's attached to this nitrogen atom that is 4 residues up the chain. And so this hydrogen bond comes between the amide bonds that we see that holds the structure together. And so alpha helices are defined by these types of what are called hydrogen bonding interactions, and that's why this is what's called a secondary structure. The entire protein isn't just a helix, but this is a portion of it that forms this ordered array. So if we start at the bottom, this is the amino acid alanine, and I know it is because the amino acid alanine has its side chain, the methyl group. And as I go to the next amino acid residue, all I have to do is move up the chain. So here's my alpha carbon. This is the one that has the side chain on it. And then I have my carbon atom, nitrogen, and this is another alpha carbon. And this one also has a methyl on it, so it's also alanine. And as I go through the sequence, I'll kind of turn this and then go to the next residue. It's alanine. Move up to the next residue, alanine. There's a lot of alanines in this. Now the next residue here is really big. That's not alanine. So we have a carbon, 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 and an amine group here. These are methylene groups, and then the final functional group is an amino group. This is the amino acid residue lysine. So this is much larger and it sticks out like this. It's considered one of the basic amino acid residues. So this one's much larger and sterically more bulky. As we move further along, we have an alanine residue, and then another alanine residue, another alanine residue, and then we come to this one. So this one's different. This side chain is an oxygen and a hydrogen. So this is an example connected to a carbon atom here. So just like alanine, this is what we call the beta carbon. There's an oxygen and hydrogen. This is a hydroxyl group. So this is the serine residue. As we move along, alanine residue, alanine, alanine, and here's another lysine residue. And then we have an alanine, alanine, and an alanine. So there's a lot of alanine residues that are in here, but once you identify the orientation of the alpha helix and identify where the alpha carbons are, you can start to rotate it and look at all the different hydrogen bonding interactions and the side chains that stick out. Again, these stick out from the going a little bit in the down direction from the sp3 hybridized carbons. So we start from the N-terminal region to the C-terminal region, and then that would mean that the last functional group we see is up here at the top with this last connection. So alpha helices show up all the time in proteins. Most proteins have them, not all, but most. Some proteins are mostly alpha helical. Globins like myoglobin and hemoglobin are mostly alpha helical. But you can't just have an alpha helix alone. Often there are just other random coil regions that connect to one section or the other. But overall, the alpha helix makes up what we call a primary, one of the primary secondary structures that we see that hold proteins together. And their structure defines their function, which is why we care about their overall shape. So this is an idealized alpha helix. They don't really look like this. They're actually bent a little bit. So while the alpha helix is a right-handed helix, it twists like this too. So this kind of looks like you could stick a rod down the middle of it, a straight bar. You can't really do that with an alpha helix. Number one, because the interior of it there's density in there, but the thing isn't perfectly straight. They all curve a little bit. So no, there's not much you could say about the symmetry. The hybridization though matters. These carbons are sp3 hybridized and the amide bonds are sp2 hybridized so that they're flat, and that contributes to the rigidity of the structure. I guess I didn't mention that. It looks symmetrical, right? Like if I do this, it looks like it's got C2 symmetry kind of, but it doesn't really. Another feature about the alpha helix is that it is amphipathic. And what that means is that one side of the alpha helix has a different type of polarity than the other. This side of the alpha helix, when I point it this way, has the lysine and the serine residues pointing in this direction. Now, the lysine residues have amino groups on them that are good hydrogen bond donors and acceptors, and the same is true with this hydroxyl group. This is the hydrogen bond donor with the hydrogen, and the oxygen can accept hydrogen bonds. So this side of the protein can interact with water a lot better than this side can, where we just have the alanine residues. These are all hydrophobic. And so an example of that would be like in this 3D printed model of myoglobin. There's an alpha helix that's seen here that is on the surface of the protein structure. So the way this is printed, this is an example of just what we call the C alpha trace. So there's no side chains that are shown here. These are what we call the backbone atoms. So in this model, if you removed all of the side chains that I pointed out, that would basically leave behind the backbone atoms. And the reason why we do that is because if we included the structure of all the side chains, these can get kind of messy and difficult to produce. But this shows the overall what we call carbon skeleton, kind of like in our bodies, an X-ray shows our skeleton. We know there's skin and organs surrounding them, but sometimes looking at just a skeleton is helpful because those other things are in the way. And so what this is showing is that if we have an amphipathic helix that's here, the amino acid residues that would be coming off this side of the helix are pointing in this direction, and myoglobin is a very water-soluble protein. So these on the surface are allowing it to interact with the solvent surrounding it, but the residues that are on the inside that face toward the core of the protein are going to be hydrophobic. And the reason why is because if they weren't, this alpha helix could literally peel away from the inside of the core of the protein would unfold, and that's called denaturation. And when proteins denature, they can't do anything anymore. They can't maintain their function if they don't hold together. And when it comes to proteins, the hydrophobic core of the protein is what holds it together if it's soluble so that it doesn't fall apart. So things like amphipathic helices are actually very common because helices like this often show up on the surface of the protein. One side has to be able to interact with water, and the other side has to be hydrophobic to allow the protein to fold and to stick together properly.
We all know that you really liked that video, and now you know a lot more about the alpha helix. This YouTube channel is dedicated to animations and videos about chemistry and science in general. So check us out, like and subscribe.